The CRISPR-Cas13 system can programmatically bind and cleave RNA, which was originally identified by the Fengzhang lab in 2016. Cas13d is a subtype of the Cas13 system, originally identified by Konerman et al. And Yan et al. Because of their small size and high targeting efficiency, we used Cas13d subtypes to develop PACMAN. PACMAN is a pan-RNA viral inhibition strategy that harnesses as few CRISPR RNA guides as possible to target RNA viruses across the same family, even emerging viruses that lack an effective vaccine or other pharmaceutical. In this video, we'll show you the key features of the PACMAN CRISPR RNA repository. The two guide pooling features are the group-specific guide RNAs and the family-covering guide pools. Let's start by exploring the SARS-CoV-2 focus page to learn about our previous research and how these CRISPR RNA were validated. Type crispr-pacman.stanford.edu into your browser to get to the home page. Click on the yellow bar at the top of the page. You can visualize the guide pool of CRISPR RNAs targeting SARS-CoV-2 on the UCSC Genome Browser by clicking the link under the first tab. Select Cas13 CRISPR under Mapping and Sequencing. Each guide was filtered and ranked based off of its coverage of the viral genomes in the group pool and its efficiency score. Let's click on the third tab to learn more about the score. In our research, we were able to show that this efficiency score has a high correlation with the reduction of SARS-CoV-2 synthesized sequences in vitro. You can click here to see the correlation for guideset 1 and guideset 2, respectively. These reproducible results suggest that the score predicted by Wessels et al. Is suitable for predicting the ability of Cas13 guides to repress SARS-CoV-2, and the score can be cautiously expanded to evaluate guides against other RNA viruses. We use this efficiency score to predict the inhibitory effect of the computationally generated guide RNAs. Under the second tab, you can download a custom set of SARS-CoV-2 guides. Click the pink Download Guides button to download the guide sequences with at least 50% coverage in TSV format. Click the SARS button in gray. The chart adapts to show the number of guides that target SARS-CoV-2 and SARS. And you can also click on specific genes to specify which genes you want the CRISPR RNA to target. Check the slider for high-efficiency guides. A high-efficiency guide has at least 90% coverage with 0 mismatches and an efficiency score of at least 0.5. You can download any selected subset of guides by clicking the pink Download button. Now let's learn about the PACMAN CRISPR RNA designs for both the group-specific guide RNAs and the family-covering guide pools. Let's click on How it works in the top left corner to learn more about the intuition behind the PACMAN CRISPR RNA design. The group-specific guide RNA design creates an extensive set of CRISPR RNAs with at least 50% coverage of all viral genomes within a group. As a complementary method, the family-covering guide pool design tries to find the smallest set of guides that effectively targets all viral sequences in a single family by using a greedy algorithm that tries to avoid excess overlapping targets. In both designs, guide RNAs that align to the human transcriptome with fewer than 2 mismatches are filtered out of the guide pools to reduce off-target effects. Finally, let's move on to the two main features of the CRISPR RNA repository. Let's focus on the group-specific guide RNAs feature of the repository first. Navigate back to the home page by clicking Home in the top left corner. Make sure that the group-specific guide RNAs block is selected. Scroll down. Notice that the group-specific guide RNAs are organized by the family of virus. The genera in red are viruses that have caused epidemics in the past, while the genera in blue are subsets of all viruses that are human infecting within that specific viral family. Choose HIV under HIV-1. This diagram is a second example of what we already saw on the SARS-CoV-2 focus page. It shows the number of guides targeting a specific nucleotide region, the genes associated with each nucleotide region, and the relative levels of conservation throughout the viral genome. You can download this pool of CRISPR RNAs in TSV format by clicking on this link. For a complete list of the HIV-1 genomes that were used, you can download the list of accession numbers in text format by clicking here. As before, you can specify for a specific subset of guides by clicking on certain genes and choosing to check the slider for only high-efficiency guides. By clicking the pink Download Guides button with the slider selected for only high-efficiency guides and the GAC pool gene selected, you will download only the high-efficiency GAC pool targeting guides in TSV format. Now let's explore the family-covering guide pool feature of the repository. Go back to the home page and select the Family Covering Guide Pools block. Choose Filoviridae. First, let's explore the phylogenetic tree. The colored areas that make up the tree itself represent the different genera of virus within the Filoviridae family. These colored bars outlining the tree show the coverage of the different mini-pool CRISPR RNAs in respect to the viral genome sequences. The outer gray bar represents the viral genomes from the repository that are human targeting. For an interactive version of this phylogenetic tree, click this link. If you click the second tab for mini-pool guides, you will find the CRISPR RNA sequences that target viral genomes in the Filoviridae family, along with both the cumulative number and the cumulative percentage of viral genomes that the CRISPR RNA sequences can target with 0 mismatches. Notice, for example, how just 5 CRISPR RNA can collectively target over 99% of viral sequences in the Filoviridae family. And this is illustrated in the phylogenetic tree as well. You can download this guide pool of CRISPR RNAs in TSV format. You can also download a dictionary of guides and the accession numbers of the sequences they target in JSON format. For a complete list of the Filoviridae genomes that were used, you can download the list of accession numbers in text format by clicking here. Congratulations! You now know how to utilize the PACMAN CRISPR RNA repository to apply your database of CRISPR RNA guides to your own research. And if you would like to contribute to our repository, please feel free to visit our GitHub page. Thank you for your interest in the PACMAN CRISPR RNA repository and all contributions.